Among male patients diagnosed with cancer, those with a prior prescription for common PDE5a inhibitors, drugs conventionally used to treat erectile dysfunction, exhibit higher long-term survival rates. This observation emerges from an analysis of electronic medical records provided by Clelite Health Services. By tracking patient data across colon, lung, and prostate cancers over 20 years, researchers isolated a survival advantage tied to these specific medications. To isolate the signal, the data was matched for potential confounders. Even after controlling for body mass index, pre-existing comorbidities, and baseline cardiovascular medications, the survival association remains statistically significant. PDE5a inhibitors are designed to promote vasodilation by relaxing smooth muscle tissue. However, the breadth of this cancer survival data suggests these drugs are interacting with a systemic biological vulnerability shared across multiple cancer types. Demographic data identifies the anomaly, but identifying the cause requires moving from population statistics to in vivo models. Validation began with the 4T1 murine breast cancer model, a cell line selected for its aggressive and predictable metastatic profile. When the mice were treated with sildenafil, the primary tumor data presented a contradiction. The drug did not reduce the mass of the main tumor. Primary volume and weight remained unaffected by the treatment. But these bar charts reveal a contrast. While the primary tumor mass remains stable, the count of metastatic lung foci in the sildenafil-treated cohort was significantly reduced. Testing vardenafil, an alternative PDE5a inhibitor, and utilizing genetic shRNA to directly knock down PDE5a expression perfectly replicated this restricted metastatic phenotype, confirming it was not an off-target drug effect. The data suggests inhibiting PDE5a does not target the bulk tumor. Instead, it cripples the cellular machinery required to execute metastatic migration. To find the molecular mechanism driving this metastatic paralysis, researchers performed RNA sequencing on the sildenafil-treated cancer cells. This volcano plot reveals a paradox. Sildenafil upregulated a specific cluster of genes driving cholesterol biosynthesis. At the top is SREBP2, a transcription factor typically activated only during severe cholesterol starvation. Mass spectrometry lipidomics revealed that while production machinery is high, the actual levels of free, bioavailable cellular cholesterol drop significantly following treatment. Simultaneously, esterified cholesterol spikes. The lipid is present but physically sequestered inside the cell and unavailable for the membrane. Sildenafil induces a state of localized cholesterol starvation. The cancer cell senses this deficit and hyperactivates SREBP2 in an attempt to survive. The biochemical trigger for this starvation begins upstream. Sildenafil binds and inhibits PDE5a, preventing the enzyme from clearing cyclic GMP. Without the PDE5a enzyme available to break it down, the result is a rapid, saturating accumulation of intracellular cGMP. This excess cGMP bypasses traditional vascular targets and binds directly to a lysosomal transporter protein known as NPC1. The binding of cGMP physically alters the NPC1 pocket, creating a barricade that prevents cholesterol from exiting the lysosome. This failure phenocopies Niemann-Pick type C disease, a lysosomal lipid storage pathology. By artificially elevating cGMP, PDE5a inhibitors create a chemical bottleneck, locking the cancer cell's cholesterol supply inside the lysosomal vault. With its cholesterol supply trapped, the cancer cell cannot maintain its plasma membrane architecture. Bioavailable cholesterol is a required building block for lipid rafts, the sections of the membrane that operate as hubs for cellular signaling and plasticity. As these lipid rafts degrade, the structural machinery required for the cell to change shape and migrate collapses. This starvation cascade extends into the mitochondria, impairing the bioenergetics required to power invasive movement. The cell remains present at the primary site, but metabolically crippled. Stripped of the resources to move, the cancer is frozen in place. To bypass the lysosomal trap, the cancer cell relies on a compensatory mechanism, hyperactivating the SREBP2 pathway to generate new cholesterol. This reliance creates an opportunity for synthetic lethality. Clinicians can sever this lifeline by introducing a standard statin to halt cholesterol synthesis. On this metabolic map, the dual blockade is clear. Sildenafil traps recycled cholesterol inside the lysosome, while the statin shuts down new cholesterol production. Cutting off both pathways leaves the cell with zero cholesterol bioavailability, turning restricted migration into an additive antimetastatic effect. Mapping the cancer cell's metabolic escape route allows for the design of a biochemical trap built from two common, non-oncologic drugs. To confirm that this targeted synthetic lethality holds up in human biology, researchers returned to the Colite digital health records. The EMR data shows that cancer patients prescribed both sildenafil and statins demonstrated an additive survival benefit, exceeding outcomes of those taking either drug alone. This additive effect provides clinical validation for the metabolic axis isolated in the laboratory. This methodology expands the utility of established medications. By targeting host tumor metabolism directly, researchers can exploit innate vulnerabilities rather than relying solely on traditional chemotherapies. Decoding the non-canonical functions of traditional signaling molecules provides a blueprint for dismantling cancer plasticity and stopping metastatic progression at its source.